Today we will talk about the polygenic inheritance. What this polygenic inheritance is? Polygenic inheritance means simply many genes are required for the full expression of a phenotypic trait. That is, we have polygenic, poly means many, and genic means genus. That is, many genes are required for the full expression of a phenotypic trait. Many genes required for full expression of a phenotypic trait. Many genes are required for the full expression of a phenotypic trait. This is called as polygenic inheritance. The trait which is controlled by two or more than two genes, this is what is called as polygenic inheritance. So we can say that we have many genes that will constitute, that is polygenes, and they influence a phenotypic trait. Phenotypic trait. They affect, influence, control phenotypic trait. So we can say that the trait, a character which is controlled by two or more genes, this is what is called as polygenic inheritance. The trait controlled by two or more genes. Two or more than two genes. This is the case of polygenic inheritance. And how we can define a polygene? How we can define a polygene? This polygene simply means that a polygene is a gene where a dominant allele is required. A polygene is simply a gene where the dominant allele is responsible for partly expression of the trait. That means in case of polygenic inheritance, we have many polygenes. The full expression of the trait that requires all the dominant alleles of the polygenus. So we can say that how we can define a polygene. A polygene is a gene where dominant allele controls only a part of a trait. So polygene is that type of gene. In polygene, the dominant allele, जो dominant allele होता है वो control करता है only a part of the trait, whereas the full expression of the phenotypic trait is caused when all the dominant alleles of the polygenes are present. That means these dominant alleles show the cumulative effect. What is called as additive effect. This cumulative effect is also called as additive effect. That means the full expression of the phenotypic trait occurs only when all the dominant alleles of the polygenes are present. And so these polygenes are also called as cumulative genes. They are also called as cumulative genes. That means here all the dominant alleles of all the polygenes should be present. for the complete expression of a phenotypic trait. So we have got these contributing alleles, the dominant alleles of polygenes, which are responsible for the full expression of a phenotypic trait. Those are called as dominant allele, contributing alleles. What are the contributing alleles? These are basically the dominant alleles. They are basically the dominant alleles which are required, which are involved in the dominant alleles of polygenes, involved in full expression of phenotypic trait. And there are non-contributing alleles. That means recessive polygenic alleles are known as non-contributing alleles because they do not contribute to the expression of the phenotypic trait. These are called non-contributing alleles. These non-contributing alleles are basically the recessive polygenic alleles. Recessive polygenic alleles which are not required, which does not get involved in the complete full expression of the phenotypic trait. These are examples of the. Now we have got what are contributing alleles, which are non-contributing alleles. This colorator is considered as the father of polygenic inheritance. Colorator is considered as the father of polygenic inheritance, and this colorator, he worked on the quantity trait of height in tobacco plant. He studied height in tobacco plant. So this colorator, it is known as the father of polygenic inheritance. Then we have got examples of polygenic inheritance are. We have examples like skin color in humans, human intelligence. We have skin color in humans, human intelligence. Then we have height in humans. We have milk yield in animals. There are examples of polygenic traits. Milk yield in animals. We have meat yield in animals. We have cob length in maize. We have another example is cob length in maize. We have number of fruits per plant. These are simply the examples of the polygenic traits. So how we can define a polygenic inheritance? Polygenic inheritance means that when two or more than two genes, they control a phenotypic trait, this is called as polygenic inheritance. That means here many genes are required for the full expression of a phenotypic trait, and the genes whose dominant alleles are involved in the full expression of the trait, those are called as polygenes. So we can say that many polygenes are required. Many polygenes are required for the full expression or complete expression of a phenotypic trait. We have two types of alleles in case of polygenic inheritance: contributing alleles and non-contributing alleles. For example, we have gene A. It is dominant allele contributes to the expression. that is called as contributing allele, while as this recessive allele, it does not contribute to the expression of the phenotypic trait, that is called as what? That is called as non-contributing allele. And also we have studied the examples of the polygenic traits. Then we'll discuss example of polygenic inheritance in case of sicken. We have human sicken color. We'll discuss this. It is simply a quantitative trait. This human sicken color, sicken color in humans was first studied by Davenport. Human sicken color was first studied by Davenport in Jamaica, Africa. The sicken color in humans, is controlled by 3 polygenes. Sicken color controlled by 3 polygenes, A, B and C. That means actually sicken color in humans is due to the pigment that is called as melanin. And for the full production of this melanin, 3 polygenes are required, A, B and C. That means when all the 3 polygenes are present, then this melanin, it would be in an adequate, it would be in an we can say the optimum level in an individual. So we can say that this, we have in case of black, we have black, what is called as negro. This is a phenotype, black sicken color. The genotype would be AA, BB and capital C, capital C. And in case of white, what is called as albino. Then we have small a, small b and small c, small c. That means the 3 polygenes are involved in the full expression of this trait, sicken color. That means when all the 3 polygenes are present in an individual, then the individual would be black. If these polygenes are absent, then the melanin synthesis will not occur and the sicken color of the individual would be white. So we'll just, if we'll make a cross between black, negro and white individuals, we'll get the individuals with the intermediate color, that is called as mulatto. If we'll cross black individual with white, what we'll get? We'll get mulatto. What is mulatto? It is intermediate sicken color. We'll get intermediate skin color. That would be, the genotype will be capital A, small a, capital B, small b, capital C, small c. So we'll take this, we'll take parents. We'll take parents we'll take parents. We have black individual, black, what is called as negro. It is being crossed with what? It is being crossed with white, what is called as albino. Its genotype would be AA, BB, capital C, capital C. Genotype will be small a, small a, small b, small b, small c, small c. What are the gametes? This is genotype. Then we have gametes A, B, C, A, B, C, or we can say A, B, C. These are also gametes, small c. These are gametes. Then F1 generation. Then we have F1 generation, A, capital A, small a, capital B, capital C, small c. This is called as mulatto. That is individuals with intermediate color. So in the F1 generation, neither we will get black nor we will get white individuals. We will get individuals with the intermediate color, intermediate skin color. And if we'll marry the mulattoes individuals, so inbreeding of mulattoes individuals are married, so we will have F2 generation. Then we have gametes. How many gametes are formed here? We have formula, that means 2 raised power n. That means n is the number of heterozygous alleles. So we have how many heterozygous alleles? 1, 2 and 3. 3 heterozygous alleles are present. So 2 to the 4 are 8. 8 gametes are formed in this case. So we'll see how many gametes are formed. This is A, then B and capital C, A, B and small c. Then we have A, small b and capital C, A, small b and small c. Then we have small a, capital B and capital C, small a, capital B and small c. Then we have gamete, small a, small b and capital C, small a, small b and small c. These are 8 types of gametes are formed by this. We have formula 2 raised power n, where n is the number of heterozygous alleles. So 1, 2 and 3 heterozygous alleles. So ultimately number of gametes would be 8, 1, 2, 3, 4, 5, 6, 7, 8. Same is the case here. We have 8 gametes here. 8 gametes. Same is the case. We have here 8 gametes. A, B and capital C. A, B and this is capital C, small c. A, small b and capital C, A, small b and small c. Then we have small a, capital B and capital C, small a, capital B and small c, small a, small b and capital C, small a, small b and small c. So we have how many gametes? 8 gametes are also here. Suppose these are male gametes and these are female gametes, and then there is a fusion of these gametes. And we'll get what? F2 generation. And in the F2 generation, in the F2 generation, what we received, we have black, we have black, we have black, dark, fairly dark, we have mulattoes, fairly, we have fairly light, this light, and white color individuals. And in the F2 generation, we will get how many phenotypes? 1, 2, 3, 4, 5, 6, 7. We will get 7 phenotypes. 7 phenotypes. In case of, we'll get 7 phenotypes in case of F2 generation. So out of and 64 genotypes in the F2 generation. Out of 64, we have 1 black. Out of 64, how many dark? 6 are dark. Out of 64, 15 are fairly dark. And out of 64, 20 are mulattoes. Then we have out of 64, 15 are fairly light. Out of 64, 6 are light. Out of 64, 1. So in case of the ratio is 1:6:15:20:15:6:1. So this is, so these are one is black color individual, this is dark color, this is fairly dark, 15 into fairly dark, 20 that is mulattoes, and 15 is, this is fairly light, and 6 is light, and 1 is white. If we'll make a graph, we'll find that it is a bell-shaped curve. In case of polygenic inheritance, it would be like this. This is character, for example skin color, and this is number of individuals. In F2, and this is bell-shaped curve in case of polygenic inheritance. Bell-shaped curve in case of polygenic inheritance. This polygenic inheritance is also called as quantitative inheritance. It's called as, also known as quantitative inheritance. Polygenic inheritance also known as quantitative inheritance, and traits are also called as quantitative traits. So bell-shaped curve in case of polygenic inheritance or quantitative inheritance. Also we can make by means of histogram. So we have skin color, this is character, number of individuals, relative frequency in F2 we can say. Then we have what? We have more frequent or mulattoes. These are mulattoes. Then we have fairly dark. There is fairly dark. Then we have light. then we have here dark, then we have black, and this is white. So it would be like this. So bell-shaped curve in case of polygenic inheritance or quantitative inheritance. So we have here mulattoes, then this is black color individuals, these are dark, this is fairly dark, these are mulattoes, then we have fairly light, we have light, fairly light, we have light, and then white, what are called as albinos. So more frequent individuals are having intermediate color, that is mulattoes, and less frequent are rare frequents are at the extreme phenotype is rare. We can say that more frequent phenotype is intermediate. And less frequent or extreme phenotypes. Less frequent or extreme phenotypes. These are black and white, these are extreme phenotypes, and mulattoes, that is individuals intermediate color, these are more frequent. So this was the case of this polygenic inheritance, what is also called as quantitative inheritance. In case of polygenic inheritance, polygenes, how can calculate the number of phenotypes? So we have completed this. In case of polygenes, number of phenotypes, it is calculated by means of formula 2n plus 1. So n is the number of polygenes. The number of polygenes. N is the number of polygenes. So we have 2n. And how many polygenes are involved in case of second color? 3. So we have 2 into 3 plus 1, 3 to 6 plus 1, 7 phenotypes. So we have 7 phenotypes, 1, 2, 3, 4, 5, 6, 7. The number of genotypes is calculated by means of formula 3 raised power n, where n is number of polygenes. So we have 3 raised power 3. We have 3 in case of 2 to the 4. 3 are 9, 3 are 27. 9. That means 27 genotypes are formed. Sorry, here we have written 27 genotypes are formed. 27 genotypes are formed. So how we can calculate the number of in case of polygenes, how we can calculate the number of phenotypes? That is 2n plus 1 formula. That means we have n is the number of polygenes. So 3, in case of human skin color in humans, there are 3 polygenes. So 3 to the 6 plus 1, that means 7 phenotypes. 7 phenotypes in case of skin color inheritance. So number of genotypes in case of polygenes. So we have formula 3 raised power n, where n is the number of polygenes. So 3 raised power 3, that means 3 to the 9, 3 to the 27 genotypes. Here, sorry, I have written here, first I have written 64, but here 27 is 27 genotypes, 7 phenotypes and 27 genotypes in case of skin color, in humans. And the ratio is 1:6:15:20:15:6:1. 1, 2, 3, 4, 5, 6, 7. 7 phenotypes and 27 genotypes are formed. Total individuals, that is 64. Number of phenotypes 7. Number of genotypes 27. Number of genotypes 27 in case of skin color in humans. Thanks.